Hey guys, welcome back to the House of Gouldians. Today you're in for a treat. We've got a talk by Dr. Peri Bolton. So the talk itself is brilliant, and then the questions and the answers to those questions is also great. So yeah, enjoy it. It is quite long, so I would get yourself a cup of coffee and enjoy it.
I'm going to talk to you today a little bit about the work that I did for my PhD and a brief postdoc that I did. So this work has mostly taken place at Macquarie University, my PhD institution, and East Carolina University in the United States. Now I have moved on from both of those places, but I'm still kind of toddling along on some of the Gouldian stuff on the side. Today's talk. Today's talk, I'm going to have three sort of sections. I'm going to talk about the conservation genetics that I did for my PhD and the Gouldian Finch declines and what I found there. And then I'm going to talk about Gouldian Finch head colour morphs and the genetic incompatibility. And then I'm going to talk about Gouldian Finches in captivity and what I found with respect to that. So the Gouldian Finch, I'm sure you all know this, occurs across the range of the monsoon tropical savannas in northern Australia. It's experienced range declines and range restrictions like many other granivorous birds of the tropical savannas in Australia. And this came to the attention of authorities in the 1970s after finch trappers noticed a really sharp decline in numbers in the late 1970s. And so there are a number of potential causes for the decline. So the Gouldian finch is particularly in the wild, particularly fussy for certain types of grasses, and fire is a really important part of the ecosystem in these savannas. And so as the north has been developed across the 20th century, we've seen more cattle grazing and changes in fire regime, which has changed the balance of the ecosystems in terms of, the types of grasses that they like. They're also have very specific cavity nesting requirements. And so again, with more high intensity fires, that often kills trees and means that the suitable nesting cavities become more rare. And there is some suggestion that the intense finch trapping that was going on in the 1970s before it was made illegal had something to do with some of the decline that we saw, but some kind of confluence of all these factors acting together. So I'm going to talk to you about conservation genetics. So what is conservation genetics? It's basically a discipline that is involved in trying to characterize the genetic health of the species. So looking at and using genetics, we can determine certain parameters that might be difficult to determine otherwise, or just, a different measure of the same thing. One of those things is a population size measurement, so how many breeding individuals there are in a population. The number of breeding individuals is important because small population sizes lead to inbreeding. And so what happens when you inbreed, as I'm sure you all know as finch breeders, if you breed closely related animals together, if there's a recessive deleterious mutation that's carried in there, it's more likely to come up as a double recessive. And so we get inbreeding depression, which is when you've got these deleterious alleles that are coming up and leading to deformities and IQ changes and things like that. But small population sizes can also lead to the loss of genetic diversity through random effects. And so this is over the broader population. You see here, we've got a population that's 50-50 of these two genes. And then over time, because it's so small, we just lose one of the genetic variants. And this is important because sometimes these variants could be, resistance to change in temperatures or diseases and all sorts of things. So this matters when a population is small. We don't want to lose that diversity. And conservation genetics is also interested in looking at the movement or connectivity between habitats. So we can look at this on small scales, say, for example, habitat remnants, but also on the large scale. So, for example, between continents or whatever. And this is important because knowing whether we can, move individuals from one location to another in terms of reintroductions, and are they able to cross barriers like farms and things like that, or is that actually a problem? It allows us to make landscape planning decisions. So before I get into the meat of it, I just want to go through some particular figures and numbers that you might see. So I'm going to talk a lot about genetic structure. And so genetic structure or differentiation arises when gene flow is restricted between two populations. And so this little cartoon has got a brown beetle going into the green beetle, and these can be kind of visualized as genes. And so in this example here, we've got two genes that are equally distributed among two populations, and so there's 0% difference, or 0% of the variation is explained by differences between the population. And then as migration decreases, genetic differences accumulate, and so you might say there's 1/8 of the variation is explained by differences between, and so on. And this can also be visualized in this manner here. So this is a human genetic study finding clusters of genetic variation. Now, I want to reiterate that these are very low levels of genetic differentiation, but they are statistically detectable. And basically the colors represent a genetic cluster. So these European populations are slightly more genetically closely related than they are to these, I think, other Eurasian type populations. So you'll see some of these plots and some of these figures, and just so you're prepared. So in order to look at the Gouldian finch in the wild, we did some sampling in the wild. And when I say we, it was my collaborators. So this was the work of collaborators at the Australian Wildlife Conservancy, previous PhD students at Macquarie, as well as South African native Sarah Pryke. And they've sampled various locations across the contemporary range. And then we compared this with museum samples. So you can get genetic information from preserved museum skins, and we wanted to look at how much genetic diversity had changed over the 20th century during this time of supposed decline. So my PhD work focused on two types of DNA. So I'll refer to microsatellite DNA, or this is DNA that comes from the nucleus. So one copy of DNA from your mother and one copy of DNA from your father. And then also mitochondrial DNA, and that's inherited maternally through the mother. So don't worry about what these graphs are. It's just showing you what the data looks like. And again, these are the sorts of genetic data that I used. So my results, I found that the birds are connected across the range in the northern parts of Australia, all the way from Western Queensland to the western part of the Kimberley, which is it's an amazing result given the huge distances that this covers. This is just one representation of the data. So recall that other plot with the humans where there was clusters. There's no cluster. There's 50-50. Like, it's all kind of statistically indistinguishable across the range. And we tried all sorts of different methods to tease this apart, and we could not find any way that they were different from each other. Unfortunately, though, there's not a lot of representatives from Queensland, and so there is more to be said about the Queensland populations, because they have experienced the strongest declines because industry and so forth is strongest in Queensland. However, we did detect a loss in mitochondrial diversity, so the maternal genome. This is just different measures of this genetic diversity. And the most important thing to get from this figure is that we've lost about 30% reduction in the mitochondria over the course of the 20th century. But in comparison, we saw fairly little reduction in nuclear diversity over this time period. But we did see some evidence for an increase in inbreeding in the contemporary population. This part of the work, I found that overall, the Gouldian finch is genetically homogeneous. There was no differences in the genetic structure in the contemporary population and the historical population. I didn't mention that before, I apologize, but this genetic structure was repeated in the historical population as well. So there was some reduction in genetic diversity in the 20th century, but the amount that was reduced is it's not that bad, and it compared to some of the other endangered species, it's nowhere near as drastic. However, all of this is good news. We did get population size estimates that do still suggest that this bird is potentially vulnerable to losing genetic diversity due to random drift. And so care still needs to be taken in terms of managing this species in the future. So the next part, I'm going to talk about head colour morphs. Now, you're all familiar with the wonderful colours of the Gouldian finch. What you may not know is that these morphs occur together across the range. They occur at stable frequencies, approximately, from the far western side to the far eastern side, which is it's an amazing colour polymorphism that is quite rare. And broadly, we see that the black-headed morph is the most common in the wild. The red-headed morph is a little less common, and the orange or yellow head is extremely rare. Now, just a refresher for those who might not know, head colour in the Gouldian finch is sex-linked, which means it's carried on the sex chromosomes. So in humans and other mammals, that's the X and the Y chromosomes. But in birds, females have the X and the Y, or the Z and the W. And so this means that red females are much rarer than red males. So, for example, if you have a pairing with a red male and a red female, a quarter of those will be black females of the offspring. So my predecessors, so this is my PhD supervisor and the postdoc supervisor of Sarah Pryke. This is Simon Griffith. They were interested in this fascinating maintenance of these head colour morphs across the range, and so they did a whole bunch of experiments in these controlled enclosures, looking at behavioural differences between the morphs and how they kind of coexist without one morph becoming completely dominant over the other. And basically, there's a whole body of work that found that the red-headed morph is aggressive and dominant, and they in the wild they acquire higher quality nest sites, so this transfers into the wild, but this trades off against things like if there's too many reds in a small place, they spend all their time fighting each other and not rearing offspring, whereas the black-headed birds are much more chill. And so this enables them to be more common in the wild because they're a bit more relaxed and so forth. So in the process of these experiments, they were doing a bunch of interbreeding between the morphs, and they found that when you breed the different head colors together, there was a reduction in offspring survival. So approximately 30% of male offspring died, and approximately 50% of female offspring died when you bred different head colors together as opposed to the same head color. This is really unusual because this sort of incompatibility is usually seen between hybridizing different species, not members of the same species. Indeed, when we think of hybridizing species, usually what comes first is infertility. And this is a process known as post-zygotic isolation. So don't worry about the word, it just means after the babies are made, there's something that is stopping the two species from continuing to hybridize. And so this might be infertility or offspring mortality. And research has shown that essentially as two species become more and more distinct from each other, the degree to which these effects of infertility and mortality they become greater. And in particular, the X and the Y, or the Z and the W, is particularly important in this process because if you are a hybrid and you have the X chromosome from your parental species, you don't have the same chromosome of the other species to back it up. And so if there's genetic variants on there that are incompatible with the rest of the genome, it means that you're more likely to be infertile or something. And so this basically means when you see hybridizing species, often it means that females are more affected in birds, or males are more affected in mammals by these post-zygotic isolation effects. And that's exactly what we saw with the Gouldian finch. We saw that females, the heterogametic, the one with different chromosomes, were more affected by this incompatibility than the males. So a bunch of follow-up work found there was a number of different ways in which Gouldian finches were able to kind of compensate for this incompatibility. So in a series of controlled experiments, Sarah found a number of different things. She found that when you put mixed morph pairs together, the female, if presented with a male of the same head color as her, she is able to sire up to 75% of that offspring with that compatible male. So it really means that she can do a lot better out of making the best of a bad job. She can use those compatible genes and instead of her partner's incompatible genes. They also, if they don't have some of these options, they will reduce their parental effort. They will reduce egg and clutch size as well. They'll produce more sons because more females die. So it's a lot of animals have the ability to manipulate their sex ratio. And so they found that even accounting for the female mortality, they are producing more males statistically than they should be. And given the choice, they're going to mate with the same head color again. If they can avoid all of these other things that we just talked about, they can choose to mate with their own head color. They'll do that. But remember, I said that head color was carried on as a sex-linked thing. This means that in the wild, there are different frequencies of male and females of the different head colors. So you can see here we've got a lower proportion of black males compared to a larger proportion of black females. And so this means that up to 30% of wild pairs could be mixed morph. So given that we found this strong incompatibility between them in the captive birds, and that there's all these other sorts of sublethal and sort of other effects, does this mean that 30% of the wild birds in this endangered species are not producing maximally. This seems like a threatening process. And indeed, doing some mathematical modeling, some of our collaborators found that indeed this whole process of incompatibility could actually be quite detrimental. And so don't worry too much about the details, but the takeaway is that there are scenarios in which the incompatibility can actually lead to the extinction of the species completely, or for one or the other head color morph to become dominant in the population. But it also, there's a lot of parameter space where the population size is quite low. And so maybe this is part of why the population is low in what we've observed in the previous section. So we wanted to find out how is this happening in the wild, and what are they doing? How are they dealing with this incompatibility? So we had some predictions based on the work that Sarah had done as to what they would be doing. We would predict that where available, they would mate assortatively with respect to head color. So black with black and red with red. And they would do that more than just if it was random. And relative to same morph pairs, a mixed morph pair should show a reduction in the survival of the offspring, produce more male offspring. This could be a result of the sex ratio bias, but also because of the mortality. And there should be more incidences of extra pair paternity or cheating. So to look at this, my collaborators, so previous PhD student James Brazelboast and Sarah Pryke set up a nest box population in Wyndham in order to get a really kind of deep look at what was going on. No one had ever done this in the wild for this species before. They're hard to find, their nesting populations, but making nest boxes means that you can count their babies and do all these sorts of manipulations that you couldn't do on a wild bird so easily. So using these data that were collected, we found that there was no evidence that the birds were mating preferentially with their own head color. So if given the frequencies of the different head colors in the population, we can produce a prediction due to randomness of how many populations of how many pairs of mixed morph and how many same morph pairs there should be in the wild based on randomness. And we found that there was no difference from randomness in the frequency of different pairs. So they're not choosing to mate with the same head color. They're just kind of doing it at random with respect to head color. James found that there was a slight tendency for mixed morph pairs to have lower offspring survival, but this difference was not statistically significant. So more data might show that this pattern is not real. So the second effect that we were talking about was the mortality, and there's no evidence of this in the nest at the nest stage. And what was more important was to be a red pair. So I mentioned at the start that red males are able to source the best quality nesting sites and are able to fend off other competitors better than the black. And so presumably there is some kind of strong advantage to being a red pair, and that is the only important and statistically significant survival effect in the offspring. Similarly, there's no evidence that the sex ratio is changed. If anything, there's a slight female bias in the offspring of all pairs. And then finally, when we're looking at cheating, so we looked at this in two ways. whether or not there was any evidence of cheating and what proportion of the offspring. So recall in the experiment, up to a single mating could sire up to 75% of the clutch. So mixed morph pairs were not more likely to have any extra pair offspring than the same morph pair. And similarly, there was no difference in the percent of the clutch sired by any other male. In fact, extra pair paternity was extremely low in this species, which is unusual compared to many other bird species where they're all cheating all the time. And so just for reference, you can see the levels of extra pair paternity in the captive birds. So in the mixed morph pairs, on average, the percentage of the clutch was really quite large. And then but in the wild, that difference was negligible. So we have pretty much exhausted all of the things that we could think of to test for this incompatibility in the wild. But maybe there was some overall subtle effect that we weren't detecting. And so we looked for evidence of genetic differentiation that has occurred due to decreased breeding between the two morphs. And so we predicted there might be genetic structure according to head color morph. So using those same sorts of techniques that I showed you before, we found that in the wild, they're completely interbreeding. There's no detectable difference between them. But when we take a look at the birds from the lab that Sarah was working on, we found that there was a detectable difference between them, about 5% difference. And a follow-up study that I did found that, again, this was replicated across aviculturalists across Australia. So we have evidence for incompatibility between morphs in the domesticated population, but it seems to be absent or extremely weak in the wild, which is great news for the wild Gouldian finch. It seems to be not particularly important for their population recovery. Other processes are probably more important. But we were wondering, why did this incompatibility, where did it come from? Was it something about domestication? So there are a number of processes that occur in domestication that could be part of explaining how this incompatibility evolved. And so, indeed, when we look at the genetics of the wild and the domesticated birds, we find that they are quite strongly differentiated from each other. The differences here are in genetic both variation and genetic diversity are larger than what we observed in the declines from across the 20th century. So this is, there's evidence that they're genetically different in captivity. So what goes on in captivity? Well, we all kind of know that we've done stuff to species to make them more useful to us or more fun to be with. Like we've made wolves into these giant puppy dogs, and we've used artificial selection to do some of these things, but some of it's also unintentional. So some work in my lab, we looked at this in the zebra finch, and we found that there was evidence for adaptation to captivity, just the housing, just being housed in a different location to where they evolved. And even within the domesticated birds, there was a difference. So what we see here is that when birds are housed in indoor enclosures, they have a lower fledging success, so less birds leaving the nest than if they're housed outdoors. And similarly, given the same kind of enclosure, birds that came from the wild do much worse in captivity than those that have been domesticated for 50 years or however long they've been in captivity. And so this is not necessarily a deliberate act, but there is adaptation to captivity going on here. And indeed, if you look at the entire genome, so this is just a representation of the zebra finch genome. Some of these little points are potentially genes that are under selection that we found there's evidence that they've changed in the domesticated bird. And one of these genes is prolactin, which you might know as involved in regulating milk and milk production in mammals, but is also involved in timing of reproduction in birds. And so one of these genes was prolactin. So this is involved in changing the timing of breeding in captivity, potentially in these birds. And indeed, we also know about the very deliberate types of selection that go on in captivity. And I'm sure that's why you're all here, is because many of you are breeding these birds for show purposes. And this is a form of artificial selection. And this artificial selection, this line breeding, can lead to genetic structure within the captive populations. So in the zebra finch, we find that these white morphs are genetically distinct from the wild type morphs. We also get a loss of genetic diversity. So they get a loss of genetic diversity compared to so these are the wild zebra finches, and these are various lab populations of zebra finches across the world. And they become more genetically differentiated from their progenitor as well as they become, as they lose genetic diversity. But also they become differentiated from each other. So these were birds that were sourced from across the world. But as you can see, these clusters from different continents they're clustered separately and not all together, so suggesting that these populations are potentially evolving separately. The other thing that happens in domestication is inbreeding. So, for example, inbred zebra finches have a different bill color, different song rates, and more abnormal sperm and fewer motile sperm than outbreeding birds. And so this is just one example. And sometimes we want inbreeding, sometimes we want inbreeding to bring out certain colors and things like that. But there are unintended consequences of this. And indeed, the loss of genetic diversity and the kind of inbreeding that goes on in the domestication process means that there's a cost to domestication as well. So many of you who may have been involved in particularly purebred dogs, they have a whole suite of health problems, and this is owing to the domestication process. Genetics has found that domesticated dogs have more amino acid substitutions in known disease genes than their wild counterparts. And so domestication has caused these dogs to be just unhealthy. So this was all what's going on in domestication more broadly. And then now we want to sort of bring that back to the Gouldian finch. What do we think? How do we think that this is involved in the head color incompatibility? Well, we have some predictions about what we expect to see about what's going on in the domesticated birds. So we expect to see a reduction in the genetic diversity in the domesticated birds, and also restriction in gene flow between regions. And particularly important is evidence for a different selection regime on head color in the domesticated population. And that's one of the key points for this incompatibility. So again, we have these wild samples that I talked to you about before that we had for my PhD. And I followed that up with collecting feather samples from finch breeders from across Australia, and looked at the mitochondrial and nuclear DNA of those. And from those, we found that there was a 32% reduction in the nuclear genome. Now recall, in the wild birds across the 20th century, there was no reduction in nuclear genetic diversity, and 9% reduction in heterozygosity, and is paired with a really strong increase in the level of inbreeding based on heterozygosity. And then similarly, we see large reductions in the mitochondrial DNA as well. Almost 50% of the genetic variation has been lost in the Australian domesticated populations. And this because we were trapping all through the 1970s and 80s. This is probably the largest and most diverse population of Gouldian finches out there. We also found evidence for restricted gene flow. So we can see that we found these clusters that are kind of come out according to region, but differences among breeders is much more important. So 5% of the genetic variation is due to differences among breeders. And so this is just owing to they're not exchanging birds with each other as much. And so there are strong line breeding and family effects going on here. So there are strong family groups and they're not sourcing out with their neighbors very much. And so that's why we're seeing these sorts of effects. And then the final step was to look at the effects of selection on head color. Now, to do this, we found the gene that was involved in head color. And so we tested all of the wild birds and all of the domesticated birds for this head color gene. And we found that, now I know this is a complicated plot, but the most important part is that there was in the wild, this is the black males, we see a decrease in the frequency of black males in the domesticated population. And we see an increase in the frequency of homozygote red males in the domesticated population relative to the wild. So this is all kind of interesting. We know that I paired these results with a survey, and I know that about 40% of finch breeders in Australia keep their morphs completely separate, and so that could be explaining some of it. I know that there is some also desire to show for red males, and so by line breeding for red males, we're going to see an increase in homozygote red males. But all of this is to say that the evolutionary processes that are going on in captivity are different from the wild, and therefore this may explain the results, the evolution of incompatibility in the domesticated populations, but not in the wild populations. So to summarize, we found low population size in the wild, but it wasn't catastrophically low. People need to pay attention and keep managing, but it's not like they're on the brink of extinction. We found no evidence of genetic incompatibility between head color morphs, which was great news. And we found the start of some evidence for a role in the domestication process in generating this morph incompatibility that Sarah found. And we're going to follow this up with cool genomics work. So we're going to sequence the entire genomes of wild and domesticated birds to get a really focused in look about what genes might be involved and what's different between domesticated and wild birds. I am just standing on the shoulders of giants. This work was funded by many organizations, as well as helped by many collaborators. And I want to thank all of them. And do we have any questions?
Doctor, thank you very much for that talk. It was really informative, and thank you so much for sharing your valuable knowledge with us. I think it's absolutely amazing. Just the first question really relates to diet. In captivity, I found of the years of keeping Gouldians, I've kept them for about 30 years, maybe more, 35 years or so. I found that protein levels play a role in determining the sex of the offspring. So simply put, when I increase proteins for the birds, I had a higher ratio of hens or females popping out. Did you find similar things in your research where diet played a role?
I don't know. So that would have been Sarah that did that work and not me. I don't think she did terribly much in the way of diet manipulations. But I would say in terms of the sex ratio stuff that she did do, they would have been all fed on the same diet. Now, it's taken me back a while what that diet exactly was. I'm not entirely sure at this point. But I believe it was fairly high protein. But it was something recommended by Mike Fiddler and the Australian finch breeders for, optimal health for everyone. So that's an interesting observation. Sorry, I can't answer it directly because I don't know. But yeah, I don't think diet per se is interfering with our results.
Right. No, thank you. Sorry, it might have been a bit of an unfair question, so apologies for that.
No worries.
And then just a further follow up to that, it's also relating to I won't say diet per se, but us as breeders, we often are our own worst enemy. So we're so fixated on colors and outcomes of certain breeding types to get a silver or a blue or this or that, but we neglect the feather quality of the birds. And what I'm starting to see is the feather quality being impacted by these so-called cross breedings and so on. We're starting to see a harder feather, so to speak, like the chicken industry. Did you guys notice any differentiation in plumage between the wild types and the domestic types in terms of the research you did? I don't know whether your research actually covered the physiology of the bird as well.
Yeah, no, as far as I know, we didn't go into a great lot of detail into feather physiology. So I wouldn't be surprised. Also, the samples that I got were almost always wild type birds where possible. So I tried to avoid the effects of having blues and yellows and all of the color morphs in that population. So I don't know. I know from my other work that I've done since that feather color and feather structure can actually be quite intimately linked. So, for example, in the mannequins that I've been working on, feather color changes the structure in such a way that they might actually be less good at water shedding in particular colors. And so I'm not surprised that you found differences, and I would expect that probably these non-wild type colors would be more affected. And that, yeah, obviously also inbreeding on top of that is probably also going to have all sorts of effects as well. So, yeah, I mean, I would probably recommend just throwing in your friends' birds every so often into your line just to keep things fresh.
Thank you. And then I'll one last question, and then I'll give somebody else a turn. One of the things breeders suffer with is obviously parental skills of the parents, and that's again due to breeding the way we do. And I was once given a lecture by an old breeder. He spoke of a term called fecundity, which is basically the breeding capacity, if you like, or the mothering skills of the parents, or whatever the case is. Do you think it is possible to breed that sort of skill through your line, or is it a random thing?
It's definitely breedable, and I mean, I think the one of the figures that I showed, potentially speaks to that. I know it's not exactly the same measure, but potentially these domesticated zebra finches are more fecund in this environment than the wild ones. Whether that is how you would breed for that, I'm not entirely sure. I mean, I would say I would pick, pairs that are consistently producing lots of offspring and keep letting them do that. One thing that's really important that we didn't kind of go over is free choice in pairing. And so we found with the zebbies, if you just kind of let them choose their partners, they do a lot better than if they're forced to be. I mean, that makes sense, right? If you're forced to make a baby with someone that you don't really know, you don't really get along with them, that's not as happy as if you're letting them choose who they want to breed with. And so I would say you can, to a certain extent, actively select for that by picking, when you find pairs that do well, promoting those at the expense of others. But also, I know it's hard when you want to line breed and stuff like that, but letting them choose their partners is also, I think, important when it comes to offspring success.
I've found that as well. So that's spot on. I think in every environment, I found two things happen. One, the results and the numbers of chicks raised are far greater when they self-select. But the other thing that happens, which is really interesting, is that there seems to be a very hardcore pecking structure. So there'll be an alpha pair or maybe an alpha and a beta pair that will dominate the breeding and it won't allow the others to do anything. So that's also another.
Yeah, I wonder. I mean, that might be a function of the space. I mean, obviously there's always going to be some kind of hierarchy, and the Gouldian Finches, the red pairs, they're the bosses, at least in the wild. But I wonder if you were able to provide more nest boxes, and I know this is not necessarily possible, given we're working in backyard kind of situations, but lowering stocking densities or providing more nest boxes and things like that might help a little bit with some of that pecking order stuff.
Yeah, no, it does. And in the experiments that we've done or I've done, I found the optimum ratio is 5 pairs, and the minimum ratio is 3 pairs. As soon as you go below 3, there's incessant fighting, and as soon as you go above 5 pairs, same scenario. Which is interesting, and space plays a big part as well, because they need a certain square meterage per pair, and they defend their territory quite rigorously. So that's interesting. Thank you.
It's interesting that they do that at low densities as well. I wonder why they're fighting at low densities.
Yeah, it is interesting. It's probably just instinct or whatever kicking in, but it's just an observation. So thanks, Doc. I'll park it there and let everybody else have a go. Thank you.
No worries.
Okay, so you mentioned decision around the offspring. Well, not conscious decision. You said that there's a predominance towards males because of survivability and that kind of thing. And that's where I was going with it. Is there any kind of ideas on what is the mechanism that that enables the hens to determine that?
Enables the hens to determine having more male offspring?
Correct. On the redheads, you said that because of incompatibility.
Yeah, so that's both red and black. So red and black females can both do that. We know that there's all sorts of structures inside a lot of female birds that are able to help with sperm filtration. And so we don't know exactly how it works. But we know that, there is what we call cryptic female choice. So females are somehow able to select among sperm from males. So, for example, in that when they're, they've got a husband essentially, and they're mating with one other guy once, they're able to, presumably they're mating with their husband many more times, so there's a lot more sperm, but they're able to choose the sperm from this one guy over preferentially over the husband. And so there's all sorts of structures and chemical things that are going on inside female reproductive tracts. There's sort of storage places, and every species is different, and we're sort of only kind of just starting to unravel all of these structures and how they might work. But yeah, I suspect it's something like that there's something going on internally where maybe there's a receptor on the cell on the outside of the sperm of the same color, and just somehow that means they're got an easier time traveling up the rest of the way, or something like that.
Yeah, so it'd definitely be sort of a physiological aspect that's at play. Yeah. In terms of that, I'm understanding.
Yeah, I don't know that they're consciously sitting there thinking, all right, reproductive tract, I want this to be happening. But yeah, I mean, whether the stress hormones that they're with the wrong mate, and that causes a physiological cascade which creates conditions that might enable certain types of sperm to reach the egg preferentially or not.
Okay, so stress hormones could be a play, and okay, interesting. Yeah. Do you think, touching on what Victor was implying with regards to protein levels, do you think maybe there could be differences in head color in terms of food availability, where because they're more dominant, they're getting first choice at certain proteins based foods and chasing others away? Do you think that could be an aspect?
Yes, certainly in mixed enclosures that would be happening, I imagine, based on the dominance hierarchies that Sarah found. All of the experiments that she did on this incompatibility and stuff like that, they were single housed. So they had the same food. There was no opportunity for interference. I suppose in a mixed morph pair, there might be interference from the other partner. So a black with a red, the red might be bullying the other partner into getting less food. And that might be having an effect. It's also possible that there is maybe you guys are probably well placed to test this, that we do know that, because there's all these other traits that are associated with head color, why not ability, protein requirements and things like that. So potentially there's just different requirements, diet requirements for the two species, or one morph is just doesn't absorb it in the same way or as well. So some of that could be affecting these things as well. Yes.
Okay, interesting. And then I think this is relevant particularly to us as captive breeders, but we are our own worst enemies, as Victor was saying, in a lot of regards. And one of them is that because we're quite competitive on the show bench, we sometimes want to keep our best birds to ourselves and that kind of thing. And particularly certain species, they have very low numbers now left in captivity. So South Africa has fairly sort of tight import controls, and it's not that easy getting new blood into the country from elsewhere. Yep. So we kind of a limited population. I'm not saying it's impossible, generally speaking. So the question is kind of, at what point number-wise are you in trouble? Let's put it that way.
That's a good question. Yeah. So, I mean, the sort of the recommendation is you kind of want to have approximately 50 breeding pairs to stave off the worst effects of inbreeding. And these 50 pairs, have got to be, or 50 individuals that are breeding, and they've got to be roughly breeding equally. So if you've got line breeding going on, say there's 50 birds, but they're all only breeding with each other and not breeding with another breeder or whatever, then that you're effectively making your population size smaller. It also depends. So some species are just better, not adapted, but like have been inbreeding for so long that inbreeding in captivity or inbreeding in the endangered context is less deleterious than a species that was previously extremely common and has declined really rapidly. So if you've been breeding inbreeding for a really long time, we have this process known as purging. So it means that these nasties, selection kind of weeds them out after a while, and so eventually, we don't get so much of these nasty things going on. Obviously, that's not something to be experimenting with, and this happens over longer timescales than I think we can really operate with in breeding. But that is to say that genetic background does play a role there, and so if it is something that's come from a historically small population, say it's a rare bird in the wild, rare bird in captivity or something like that, and it's rare in South Africa, that's bad because you might lose them just to a random event. But on the other hand, it might be not as disastrous as, say, 2 zebra finches left or something like that. If you've got the zebra finches come from a huge population, and so a small population of zebbies might be worse.
Okay.
As everything in science, there's no one answer, I'm afraid. But
fair enough.
Yeah, I would say 50 is kind of a good number to be thinking about.
Okay. Then one of your sort of research interests was the behavioral genomes and that. I was curious. With me, I did psychology as well, so behavioral aspects, we talk nurture versus nature. I was just wondering sort of what other aspects besides maybe the courtship dance and that you've become aware of that is definitely sort of genetically inherited opposed to sort of being taught. We touched on pairs being better parents, so you said we can breed for that, but is there anything else you've observed or comments you'd like to make on that?
I don't I mentioned to you in the chat, I haven't I did a lot of this because there were samples already existing, so I have not personally observed something differences due to things like courtship displays or anything like that. Detecting whether something is nature versus nurture is actually somewhat difficult to do. Because if you learn something from your parents, that can come up as can be sort of seen as inherited, even though it's just learning, and so there is some kind of interaction there. But we do hope, and there's no such thing as like a single gene for, say, courtship or anything like that, but we do hope that this whole genome study will be able to find genes that might be involved in regulating breeding. So it's not necessarily that they say, well, this guy's a good breeder and this guy's not, but it might mean that this guy is 20% a better, might 20% be slightly better than his neighbor, and that gene contributes 20% of his better at breedingness compared to this other genetic variant, things like that. So there's subtle effects that any genes that I would find would be having.
Okay, interesting. And then I know once again when you and I were chatting, you mentioned it wasn't really your side as that, but with you sort of looking at specimens in the lab, was there any kind of locality differences that, other than head colors and that, but that's not really locality. You said that's those numbers are stable across the range. But is there anything that you did observe that tended to be from certain regions or not really?
Yeah, so no, not really. I mean, I didn't exactly partition the data that way. I did look at size and things like that with respect to head color morph, but nothing that I've read or nothing jumped out at me as like, oh, the birds from Western Australia were so much larger than the birds from the Northern Territory or something like that. There wasn't anything like that. And as far as gross plumage traits go, they're all kind of the same. There might be subtle differences, but they're hard to find in the wild. They're hard to get your hands on. And so no one's really gone out and really gone into great detail about these things. And yes, there's some museum specimens, but the work that has been done didn't show anything on those.
Okay, interesting.
Yeah.
It's just one of the sort of aspects that was up or being debated recently was I don't want to sort of sway you in either way, so I won't mention which was which, but on the one side of the standard, they don't like a fusion bar because they say that it's a fault through mixing of certain structure and color pigment and that. But on the other side, they're saying that the fusion bar should be there, and they actually encourage it on their show birds and they want it there. So that's where I was kind of going with it, if you had observed any locality differences, because I thought maybe, the one
What's the fusion bar?
It's the line that separates the yellow on the belly from the purple of the chest, basically. So it comes across as a red line. But what sort of counteracts the idea about it being a structural fault, if you like, where there's pigment where it shouldn't be, is the fact that the white-breasted Gouldian, should I say, you also get a fusion bar. So if it is a mixing of the purple and the yellow, the lutein of the belly, you would think that it would only be purple-breasted birds that have it. So yeah, that was where I was going with the locale question.
Yeah, I don't know if anyone's looked at it in that great detail in the wild, and I don't know, honestly. I don't know that it's very common in the wild either.
Okay.
So it might just be there occasionally, but I don't know that anyone's really looked at that trait in particular.
Okay.
But that's not to say that someone couldn't do that. Like the museum specimens are there, but yeah, I don't know.
I know that kind of locality differences, like corn snakes, for example, is prevalent. They've found that there's definitely differences between population groups, depending on where the corn snake comes from. I think the Oketees, I stand to correction, is an example of that, where there's a lot more black in it. I think that is a sort of a locale subspecies, if you want to call it that, versus others. So yeah, I was just wondering but you've answered the question, so thank you.
Yeah, no worries. I mean, I think probably one thing that would be kind of explaining this sort of no one knows about any obvious differences. There might be subtle differences, but I think because they're exchanging genetic material across the range, there isn't a lot of opportunity for these traits to kind of diverge either. So I mean, we don't know about in far Cape York or anything like that. They might be a bit different, but as far as we can tell, they're kind of all the same thing. They're all interbreeding freely, that the same gene over there in Western Australia is the same genes in the population in Queensland. So yeah.
So if I'm understanding you correctly, basically the differences might exist in the wild, but because they're intermingling all the time, they don't have a time to sort of perpetuate and come out. I'm talking now something that's not necessarily going to increase predation and get taken out, like a blue wood, for example, or a yellow. But in captivity, because we're now focusing on something, it then has that opportunity to develop, because now we've got isolated gene pools. Would that kind of be an accurate?
Yes, exactly. And that's sort of how the speciation process works, and how this geographic variation arises, is that there's restriction in gene flow. Sometimes there's adaptation as well involved, and so sometimes they're exchanging genes, but they're just so adapted to a particular environment that they have to look different. but this, I mean, more or less they all live in the same environment. They're all kind of the same population. So yeah.
Okay, no, great. Is there anyone else that has any questions?
Yeah, I think Victor has another question.
Okay.
Oh, thank you. Mute button playing up again. Just two questions really. The one is related to your observation around the results of breeding from head color combinations, and I found that absolutely fascinating. But for me, what immediately sparked a thought in my mind was you noted in the wild there was no preference for head colors, and I wonder if it had to do with the fact that birds see in the ultraviolet spectrum. So what we see is not necessarily what they see of each other, and maybe the selection is more around vigor and quality of the pair, the partner, I suppose, vis-a-vis the actual head color itself, because as I say, they see in the ultraviolet spectrum. So it's more of a comment than anything.
Yeah, absolutely. That's what we said in the paper as well. We said, like, it seems that color is not important. This particular visible spectrum of color is not important, but there's all sorts of other traits that they could be selecting for, and ultraviolet is one, but they could also be just selecting, maybe they like fatter birds.
Yeah. No, it's quite possible, because the bird could represent a better partner for feeding and raising the young and so on.
Exactly.
Absolutely. Yeah. The question I want to pose to you, Doc, with the silver and blue birds, more blue than silver rather, there's obviously a lack of the in the carotenoid side of things, there's lack of lutein, the yellow.
Mm-hmm.
And the birds tend to be weaker and smaller and all of that. Do you think, in your view, the lack of lutein has a part to play there? Because there's papers and research that have found lutein is linked to immunity and to improving the overall bird's health or the animal's health. Do you think that has a role to play in that?
Yeah, that's a great question. It's entirely possible. We know all about, we want to eat carrots for the carotenoids and things like that for our health. Absolutely, that could be part of it. We also do know, though, that they are able to turn on and off. So with gene regulation, we're all able to turn processes on and off in different tissues. So that would also presuppose that they can't process lutein in their entire body, whereas the mutation could just be affecting their feathers. So it's possible, it's absolutely possible. But I don't know how their physiology is working. The other possibility is that this is a result of the line breeding and other unintended effects of selecting for this particular color that is causing them to be less fit than the wild type birds.
Yeah,
I can't answer that, unfortunately, but yeah, either hypothesis is good, and that's something that someone one day might test.
No, it makes sense. It makes sense, because what we try to do as breeders is not necessarily breed two like birds together. So we won't breed a bluebird with a bluebird, because that just I suppose it exponentially worsens the bird's health and size and vigor and all that. So typically, what we would try and do is pair that bird back to a normal wild type bird, and then the outcrosses from there back to the mutation, so that you try and build a stronger base, I suppose.
No, that's good. Yeah. The other thing is, like, if you think that the carotenoid is is a digestive problem, or unable to process it, the other thing is that maybe you could try doing some carotenoid supplementation. So maybe they're just less good at processing it, so maybe they still can, they're just not as good. And so maybe if you supplemented, gave them feed that have higher carotenoid levels, they might do better. That would be something that you could test.
That's a good idea. I need to try that. Thank you.
for the benefit of the guys on the call, if as a breeder, if a layman's sort of version of experimenting, I've for our own club, I've sort of suggested the guys, if they're going to introduce anything new, so in this case, a diet difference or something like that, is to try and separate into different groups so that you've got a control essentially. But maybe if you can just give them a couple of pointers on sort of how to go about it. So what is a sort of a minimum sample size you want to be using, for example, to get a worthwhile result? I know we're talking about now the statistical aspects, and there's differences between groups and correlation coefficients and all the rest of it. But kind of as a bare minimum, as a layperson, what are we talking?
I mean, the more the better, the more that you can do, the better. It does depend on, what kind of effect you expect to see as to what kind of sample size you need. So if you expect the effect to be small, then you need a very large sample size to demonstrate it. Whereas if you expect the effect to be large, then you need a smaller sample size. But the more the better. So, I would say, probably standard behavioral ecology kind of experiments have like 10 individuals in each treatment. The other thing that you might want to think about is, it depends on what resources you have available, different aviaries with, different other configurations. So like if you've got sun on one aviary and sun not on the other aviary, or like 4 aviaries that 2 have sun and 2 have not. So I would have one of the sunny aviaries with extra carotenoid, and one of the shady aviaries with extra carotenoid, and then one shady aviary with normal and one sunny aviary with normal, for example. So trying to control for other things that you think that might be affecting breeding or quality or whatever.
So ideally, if there are other variables, you need to have a control group per variable type thing and a test group per variable type thing to try and eliminate and come to a better,
more or less solution. Yeah. But if you've got a setup like this where you've got small cages, you can isolate pairs or 2 pairs, and then all the other conditions are exactly the same, that's great. Or if you've just got 2 aviaries next to each other, I mean, it's not hard science, but stick more carotenoid in one aviary and the other one, and just make sure that your stocking rates and your species and whatever are more or less the same.
Okay.
It's hard to do a properly controlled experiment. So
no, fair enough. But yeah, just for Victor, if you did want to test the whole carotenoid scenario, you're saying a minimum ideal would be about 10 on both sides, on the test side and the okay.
Yeah.
All right. Okay, no, great. Yeah, from my side, just a huge thank you for giving up your time. Already, we appreciate having someone of your caliber talk to us. A lot of the time it's breeders sharing their knowledge, and sometimes that knowledge isn't quite correct, or it's misinformed. So to have, like I said, a scientist of your caliber chat to us is really beneficial to the hobby. And we really do appreciate it. So thank you.
Oh, thank you for the opportunity. I appreciate it. It's talking to the public is such an important part of the scientific process, and there's no point in doing any of this if we don't share the results. It's not just about talking to each other, it's about talking to everyone why this is interesting and why it's cool. So thank you.